So before Andreas starts, I'd just like to remind everybody we're going to, after these two talks, we're going to have a discussion session. And so this is a little time you can prepare your 3-minute burning question if you want to help us get started with the discussion. So next up is Andreas Herz.
Yeah, thanks, Bruno. Thanks for the invitation. And in some way, I think you'll see that grid cells are much simpler than you may have thought. And on the other hand, I'll also try to indicate that some phenomena are much more complicated than we think. So at the end, you're probably where you are right now. All right. And I should mention, and I do so right ahead, that it's mainly work of Martin Stemmler and Alexander Mattis. Alexander was a PhD student in the group, and Martin is a senior research affiliate. And I changed the title a little bit to reflect these issues. So we heard already from Mike quite a lot about grid cells. Unfortunately, he didn't show this slide, so I have to do it. For those that are not in neuroscience yet, so if we record in entorhinal cortex with an extracellular electrode, you will get signals from multiple neurons. And so these are, again, these firing fields. If you mark the centers of the firing fields, in this case from these 3 cells with crosses, it's still a bit confusing. But if you move all green crosses and all blue crosses relative to the red crosses, you can get them in this shape, which means, as you had only moved them, not rotated, not scaled, that at least nearby grid cells have similar spatial frequency, similar orientation, but they differ in spatial phase. So that's one key property of grid cells. And the other is that the spatial scale, it changes, and in fact it increases when you go from dorsal to ventral. So you see clearly there are different grid scales, and in fact it does not do so in a continuous manner, but stepwise. And the sequence of scales forms a geometric series. These are the main properties of grid cells I'll be basing the talk on. There are many more, but these are like the core ingredients that you should know. And that also means that in the end we can talk about grid modules. So within each module, cells obey these properties. And there are somewhere between 4 and 10 modules. This number is not settled because it's difficult to record on one end of the grid cell area, and therefore maybe it's 4, maybe it's 6, probably it's not 10, but somewhere in this range. I will not talk today about mechanisms. Yeah? This would be an entirely different talk, and there are people in the audience like Yoram and Ila that have worked extensively, and also Mike, on the various models. I will not go into mechanisms. There are these 2 different main classes of models. And one reason why I will not go into that, because I want to focus on decoding, and because neither of the 2 model classes explains why there is this stepwise increase in grid scale.
Are these different layers, or the scale that you're talking about?
They are sitting in different subparts of the entorhinal cortex, but one should be careful. There are even intermingled regions where you get different scales at the same location. So what I want to focus on is the reason why the Mosers got the Nobel Prize some years ago. And it was given to them because the grid system provides a solution to measuring movement distances and adds a metric to the spatial maps in hippocampus, an internal GPS. The last part is certainly for PR reasons. Nobody believes that there is a GPS in our brain. But there is still this notion, and you could also hear that in some of the talks this morning, that grid cells clearly offer a metric. And I'd like to question that. So is it really a metric for space, and in which sense is it? So the usual argument goes such that each cell has periodically laid out firing fields. And so, for example, if you go along that route, you will have increases and decreases of firing rate. And if you know for how long you have gone, then this allows you to measure distances. Beautiful picture. However, nobody tells the rat to move along straight lines that go directly through adjacent centers. So if the rat happens to go that way or that way, it would see a different effective scale and therefore measure with a different measuring device. 1 meter would not be 1 meter anymore. If there were only grid cells, sure enough, if the rat has also access to directional information, we heard about head direction cells earlier, it can put this information together and compute distances. But using grid cells alone, it's difficult. Using a grid cell alone, I should say. And in fact, nobody tells the animal to move along straight lines. It could also go in circles, come back to the same position, have the same firing pattern, but not moved at all. So, what can we say? If you go on and speak about spatial location, whether the grid cell system can be used to do that, then you run into the following question. And I have done it in a idealized form. These are 3 grid cells, and they have aligned orientation, similar grid scale, but there is a spatial phase shift. That's grid cells within one module. And you may wonder, why are they arranged in that way? Because obviously, because they repeat periodically, the readout does not know whether the animal is here or 1 period away. There is no information in this periodically repeating pattern. And that's slightly puzzling, because if the grid fields of different grid cells within one module were rotated with respect to each other, every position in space could correspond uniquely to a population firing pattern of the grid cells. And then one could decode space relatively easily. So one of the questions I want to address is, why are the axes aligned within a module? And there is a simple answer to that if you believe in continuous attractor models, because there all neurons are living in the same activation sheet and necessarily have aligned directions. However, from a decoding point of view, that doesn't answer this question. So I'd like to understand, does it make sense from a coding-decoding point of view to have an arrangement like that, which at first sight seems not to be ideal? And in order to address these questions, we have to think about what does it mean to encode, what does it mean to decode. And so we think about a stochastic population spike count within some spike count window. The whole population, n neurons, and their spike counts from the first to the nth neuron are given by some probability of generating this population spike count given the position of the animal. That would be our encoding model, and the decoding would mean that the animal, or we as people analyzing the data, are asking what is the most likely position x given the spike count vector at that particular point. So these 2 probabilities are related by Bayes' rule, and therefore you can, under certain assumptions that simplify the whole game, and I will make these assumptions, that there are independent Poisson spike trains, that there is a flat prior, and that there are sufficiently many neurons such that a particular term, the sum over the firing fields as a function of x, that this is constant as a function of x. This property is in fact fulfilled, or roughly fulfilled, with 10 or 20 neurons per module. So that's not a real tough assumption. The flat prior might be a stronger one, and assuming independent Poisson spike trains is certainly not doing a fair job to reality, as I'll point out at the very end of the talk. So if you put all these equations into that, you find that the posterior is given by this formula, which is compact, yet it's still complicated. How should the animal, if it wants to read out its position from the spike train pattern of the grid cells, how can it calculate this maximum? Because that's the most likely position given the spike count vector. We can do it, and in fact, when people run decoding algorithms, when they do Bayesian decoding, they do something similar. They go through all possible positions x, ask where is the highest possibility, who has the highest probability, and that's the decoded position. But how could the animal do that? How could the animal compute this horrible, depending on your emotional feelings to mathematics, this horrible term? And I will argue that under certain conditions, this can be done very easily, and we believe that the grid cell system can actually do that. And one avenue towards answering this question comes from analyzing what the firing field, actually the firing field profile looks like. And if you do so, you see that it can be well fit by what's called a von Mises tuning. That's a cosine in the exponent of an exponential function. And putting the cosine up here and multiplying it by what's called a concentration parameter allows you to scale the width of the grid field while keeping the period constant. So unlike a sine or a cosine, where the width is defined by the wavelengths, you have an additional freedom. In fact, these tuning curves can be found in many other systems. Quite often people, for example, in motor cortex, they speak about sine or cosine tuning. In fact, if they fitted the curves with a von Mises, they get much better results. So von Mises tuning is not only a mathematical hack, but it seems to really capture the firing properties of a whole range of neurons, motor cortex, visual cortex, thalamus. They're all nicely fit by von Mises tuning curves. But there is more to that. And that was the insight Martin had. And I think it's the insight of a mathematician or somebody trained in theoretical physics that you would never get if you are simply doing numerical calculations. So what Martin realized is that if you have von Mises tuning, then the posterior, which was this horrible sum, if you put in these terms, it is also von Mises. We're talking about 1D, but we can extend this to higher dimensions. And that means that the maximum of the von Mises can be calculated as a population vector, similarly to population vectors that have been calculated in the motor system. And I want to show you how that actually works graphically. Let's assume that the poor rat or mouse is sitting at a certain position. We have, for example, 20 grid cells. They fire with particular firing rates or spike counts in a certain window. And now what that means is that you arrange these spike counts around a circle and you calculate the population vector, project this back, and you'll get the most likely position. And from the spread, you also get information about how likely certain deviations from this most likely positions are. To do that, the requirement is that you can put all these neural tuning curves on the same circle. And that means that it only works if all neurons within one module have the same wavelengths. So that explains or provides an explanation why within one module we really want to have the same grid scale. That's in 1D, and if you extend the argument to 2D, it means that the directions have to be aligned, because if they are not aligned, you'll get, if you project to the x or y axis, you would get different circumferences, and then it would not work population vector decoding. So the argument goes as follows: if you want to do population vector decoding, you'd better have grid cells arranged such that they fall into classes with aligned orientations and identical periods.
Andreas?
Yes?
So, if it's only one spatial frequency, then you get a multimodal posterior. All
Yeah.
the interesting things come from fixing that with multiple frequencies, in which case it doesn't, the whole
Next slide.
argument disappears, right?
Next slide.
No, the important point is in which case the whole argument disappears. It doesn't really matter. You're going to have to do population decoding with multiple wavelengths anyway. So what is it? So it sort of doesn't seem to matter whether.
Well, if I have one module with a certain wavelength, right? I run this algorithm, I get a certain position estimate that comes with an error. And then I use the next one to refine this estimate.
So that, yeah, but you can also have sort of random phases, random wavelengths. You can decode just as well, right?
Not using population vector.
No, exactly. What do you mean? You mean not using the argmax thing?
No, argmax yes, but not this construction.
Yeah, but you can. But that construction is also not so easy for neurons to do, right? You basically. I'm confused because the middle thing holds no matter what, right?
The middle thing holds if you have Van Mises.
No, it holds in general.
Well, if you put in the Van Mises. Okay, right, yeah?
It holds in it. And the right one holds for particular choice of decoding, but you don't really need it, right?
No, what I try to argue is that if you have that, you can calculate it with this kind of trick.
But why does the brain care about that kind of thing? If you can get to that?
Because I would argue that this is doable if you have some sine and cosine wave carriers and you multiply them by the spike count, you can actually calculate that.
So it's related to the dimension as well, right?
No, this is in 1D now. And we would, if we go to higher dimensions, we would do it along, for example, rectangular coordinates.
But you have to take an arctan to do that, right? It's
Yeah.
not so easy for neurons.
Well, I mean, I'm not going into this argument whether it's easy or not. What I'm going into is that if we were able to do that, and people have argued, like in the motor system, that this is possible, then this is an approach I can follow.
Okay, I wouldn't buy the motor. I mean, but they buy. We'll talk later.
So the right side is essentially just a weighted sum of activities weighted with their tuning.
Well, Peter's point is that you have to
The middle
do.
one is really it's a complicated optimization problem, right?
Right. Well, Peter's argument is that here you have to do an arctange. And the question is whether neurons can do an arctange, right? That's essentially one of your points. But let's talk about this later, because otherwise he will kill me. So the point then would be that we can refine these estimates and come to very precise position estimates by refining step by step. Now, what falls out of that argument is also the following. Why should there be a scale ratio? And I didn't say that before. Grid scales have a ratio, the length scale, of around 1.5. Depending on the lab, it's 1.4, 1.6, but somewhere around 1.5. Why this magic number? Why does it help? If you have a single module, then the posterior would have a certain width, and you would know if you are at 0, yes, the most likely position is here, but there is a certain deviation possible. Now, if you add a second module, depending on the scale of the second module, you will, for example, for s equals 2 and s equals 1.5, you would have two situations. Let me use this first. In this case, the probability to make the right judgment increases. The probability of making a wrong judgment decreases, so that's fine. The second module helps you. If, on the other hand, the wavelength of the second module is too small, then you get these side bands. And that means that the probability of making a drastic error in your position estimate suddenly increases. You're doing much better here at the correct estimate, but you also get these errors from making large misjudgments in your position estimate. So that seems that if we stick somewhere around 1.5, we don't run into this problem, though it's preferential to have such a scale ratio.
Does that depend on your kappa?
That does not depend on the kappa, because what I plot here is the posterior.
Okay.
Yeah. Of the entire population. So we would predict that S should be around 1.5 or smaller, and that depending on whether, now for an experimental prediction, whether you silence the smallest scale module or some intermediate module, you should get very different behavioral results. If we just silence the smallest module, then resolution decreases. If we silence some intermediate module, then the remaining scale factor, if this was 1.5 and 1.5, then, and we silence this module, then this scale factor would be beyond 2, which would not be a good situation. So then we would expect that the animal makes different errors than if we could silence the smallest scale module. And there is one more advantage from mapping linear space onto a circle, which I try to demonstrate here. So far, we have been talking about allocentric location with respect to a certain origin. And I'd like to propose a framework that is somewhat complementary to what you have shown, Mike, before. I believe that this periodic representation allows us to have a way to go from allocentric to egocentric coordinates in the following way. Let us assume that we want to know, or the animal wants to know, the distance to some food source. Then what it has to do, or what it could do, is if it shifted the origin from this position to that one, then its allocentric coordinates in this new coordinate system is exactly the distance to the food source. So the question then is, how can it move the origin? And moving the origin in the spatial system is the same as rotating the origin in the circular arrangement. And now rotating this population vector corresponds to multiplying these activities with sine or cosine gratings. Now Peter will raise his hand again and say, well, we don't know how to do that. Yeah? And I do agree. I put forward this as a mechanism, but it could be implemented if we had some sine or cosine waves downstream that add into the synaptic couplings of neurons downstream of the grid cell population. So finally, I come to stuff that is not our work, but that gives a certain twist to this very simple story I presented to you. Namely that the grid patterns are not always beautiful. They are not always beautifully hexagonal, as you can see here from the data from Stensola, where clearly there are some deformations, and even stronger in a paper from Julia Kupic et al. You see that here's the same cell in a quadratic environment, here it's in a trapezoidal one, and there is no beautiful hexagonality anymore. And in fact, this result led them to claim that these results challenge the idea that the grid cell system can act as a universal spatial metric for the cognitive map, as grid patterns change markedly between enclosures and even within the same enclosure. So here they argue that because the hexagonal firing pattern is gone in such enclosures, it cannot be a metric. And I think what they got wrong is that they equated one-to-one the firing pattern with a metric. And if you think about the globe we happily live on, locally we can do nice Cartesian coordinate systems, but when we get close to the pole, things get messy. Nevertheless, there is still a metric. So just having a situation where you have distorted grid fields does not imply that the metric is gone. And in fact, what we could show is that it's still all okay if the distortions are shared from neuron to neuron. So there can be position-dependent distortions, and as long as they are the same for all neurons within a module, then you can have a readout that is not affected by these distortions. And Julia was in Munich just a few days ago. They have new results that are in press that in fact shows that the distortions are shared between neurons from the same module.
Wouldn't you need to have the same distortions also in different modules, which I think we don't know yet?
Well, they show that this is also the case. We would not. Have you seen the paper?
I don't think they showed that. They showed that it within a module.
No, in this new result.
No, they don't really show that the distortions are the same. They just show that they occur in the same locations within the environment.
No, they say it's the same. It's the same
We
scale.
can argue about what they're showing later.
Well, yeah. Let's wait until the paper is out. For our decoding, it doesn't matter because we do the population vector decoding module by module.
Right, but then when you want to put all that together, if the distortion is not the same, you would need to somehow take that into account.
Yeah, we have to take that into, but it doesn't kill the algorithm. The algorithm would be killed immediately if the distortion was different within a module. Okay, and then sure enough, the picture that I've drawn so far also is incorrect, as the grid is not as nice as you might have thought. That is by Dunn et al. And also Ismakov et al. That show that the hexagonal pattern is not always such that the firing rate maxima are identical. It may still be hexagonal, but firing rate modulations. So the simplified picture where all firing fields are equal probably does not hold. This is not a killer, however, for the population vector decoding if there are sufficiently many cells whose variations from the perfect hexagon are randomly distributed. Then you can still add sum over the different cells and effectively come back to the situation with the population vector decoding. Mike has already pointed out that there is theta and theta phase, and there is also phase precession in entorhinal grid cells. And we had looked at that in quite some detail and asked in particular the question, if you have two firing fields and you have spikes in one field and spikes in the next field, is there any relation, is there any dependence on whether a particular grid cell fires late in one field, will it fire late in the next field? And you can analyze all kinds of things, last spike, first spike, and what have you. And the result was that there is no correlation. And in fact, according to the statistical tests we did, they are independently encoding the different firing fields, which means that also from a phase precession point of view, grid cells have multiple firing fields that act as independent encoders of physical space. More to be expected. Grid cells do not only encode space, but also the contextual information. And let me finally come back to this movie, a similar one, so that is by Dori Dedekman, similar to what Mike has shown you. And this is just the last slides. And what I want to point out, and the spikes are marked with these white diamonds, and if you look at them, you'll see that the way how we describe grid cell activity by firing fields, that implicitly assumes that you have Poisson spike trains, that you can even talk about a nice firing field, probably doesn't hold, because you'll see these streaks of action potentials that clearly show that the individual spikes are not independently drawn from some distribution described by the firing field. So I think yes, and that was what I tried to get across today. Yes, there is some regularity in the grid field firing pattern that may be used to read out position, but no, there is still a lot of features hidden in these firing patterns that we have no clue about what they really mean. And what we need to do, and Yoram has done, I think, a great step forward in this direction, we also have to get away from decoding location, but remember the animal is moving. And so in the end, what we need to do is we have to think about ways to decode the movement pattern, the trajectory, and not, as we have done and many others, to decode location time slice by time slice. So that's what I wanted to tell. Thank you.
So I understand how you combine the different estimates on the different modules, but this is not sort of population code combination, right? So
No, you do population code in each level.
Bayesian inference stuff, which is then more complicated to implement than the population code.
Well, you have a position estimate on each level, and you can use this to refine on the next level. I can show you the math. Yeah.
So is there a reason to think that the downstream structures decode each module independently and have this problem, or do they just, does the downstream get the information from the different scales, and then I think, as you were saying, the problem kind of goes away, right? And the original Finn paper, before they knew they were grid cells, but they had the multiple fields, they actually did the standard decoding analysis, and they were able to reconstruct the animal position trajectory very with high accuracy. So I'm just...
So here I would say having these different modules may also buy you additional ways of encoding, because maybe the problem of decoding grid cell activity because of the multiple firing fields is only a superficial problem. If you knew at some point where you are and you know that you cannot jump, then information from one module will also do the job. It's only when you stop moving and grid cell activity vanishes, that you don't know whether you're here or one grid position or two grid positions upwards. What I want to say, you can decode beautifully if you give a start point and then use the grid cell activity within one module. Because then you don't have the ambiguity that can only arise if you either don't know your starting position or if you allow for jumps.
All right, I'll talk later. I
Yeah.
don't quite follow
If you have a ambiguous code, as long as you don't leave the range where you have clear decoding, where you have one-to-one decoding, there's no problem. So if you move along a closed trajectory, you always know where you are, even if you have a code that repeats periodically.
But that has nothing necessarily to do with the specific decoding mechanism here, right?
That's right. That's absolutely right. Yeah.
I could be in any one of these places, but I have a prior that I'm here because...
You're absolutely right. No, I just wanted to point out that the difficulties of decoding grid cells may go away as long as you know your starting point. However, if the animal decides to stop, how will the information about this location be carried? That remains a problem. But let's talk about that.
All right, thanks very much, Andreas. Next speaker is Kevin Jones.
There's no relationship. They're not being in the same phase. Different nodes. If you think of each node as a field, you're leaving, if you're between them, you're leaving one field and entering another. The firing of this one bit, I think you're saying the phase code of different nodes. Different nodes, not always